Hi all. Surely we will live with COVID 19 for some more time. We have to fight against COVID 19. There are four different ways to fight. The first is what people must do at the community level. The second is to attempt the use of convalescent syrup. The third is to look for drugs to treat the affected. And the fourth is to find a successful vaccine. At community level, we have to fight with a very strong and infectious virus for which we should work together. Always keep in mind that everyone is susceptible. To prevent the infection, protective devices like face mask should be worn. Simple cloth face mask can be used by public. It may be an easy, inexpensive and effective way to stay safe. You may wonder, why should I wear a face mask? The largest amount of viral transmission occurs early in the course of the disease. Therefore, people may be infectious even before they start to show symptoms. Up to 80% of transmission occurs from asymptomatic carriers. Widespread mask use may help to limit the transmission of the virus by people who don't realize that they may have it. Use 2 layers of tightly woven 100% cotton fabric folded in multiple layers. A bit of breathing resistance is expected when wearing a mask. Does everyone need to wear a mask? No. People who can't remove the mask on their own or have breathing issues shouldn't wear mask. Neither should children under the age of 2 due to the risk of suffocation. Here are some face mask safety tips. Wash your hands each time you put on, remove or touch the surface of your face mask. Put on and take off the mask by holding it by the ear loops or ties, not by touching the front of the mask. Make sure the face mask and the straps fit securely over your ears or behind your head. Avoid touching the mask while it's on your face. Wash your cloth mask and dry it after each use. Wash it with detergent. Keep it in a warm, dry place. To stop community spread, hand hygiene is the golden rule. Wash hands with soap and water. Clean your hands with an alcohol-based formulation if hands are not visibly soiled. It is faster, more effective, and better tolerated than washing with soap and water. Here is the procedure for hand wash and hand rub. My 5 moments for hand hygiene are the key moments when healthcare workers should perform hand hygiene. 1. Before touching a patient. 2. Before clean or aseptic procedure. 3. After body fluid exposure risk. 4. After touching a patient. And 5. After touching patient surroundings. All of you know what social distancing is. Keep your distance to slow the spread of the virus. Stay at least 6 feet, about 2 arms length from other people. Try to reduce gatherings. Limit using any kind of public transportation, ride sharing or taxis if possible. Shaking hands and hugging to be avoided. Instead, greet people with a wave, a nod or a bow. Don't touch your eyes, nose or mouth. Cough hygiene is to be practiced. That is, cover your mouth and nose with your bent elbow or tissue when you cough or sneeze. Put used tissues in the bin immediately and wash your hands afterwards with soap and water or clean with alcohol-based hand rub. Wipe your gadgets with alcohol wipes. It is better safe than sorry. So stay home and safe. Make right decisions about how to protect yourself. Know when to seek medical care. It is important to avoid contributing to panic. You can still be social by being active. Go for a walk, relax, and stay connected with friends and families. Share your thoughts and feelings. Try to reduce your screen time and seek information updates. Eat healthy, enjoy your meals, ensure that you stay well hydrated, and get a good night's sleep. The second way is by use of convalescent serum. That means serum of a recently recovered patient, which contains specific IgG and IgM anti-SARS coronavirus 2 antibodies. They can neutralize the virus, prevent further replication, and stop tissue damage. Donors must have had documented SARS coronavirus 2 infection, be symptom free for at least 14 days, and meet standard blood donor eligibility requirements. Before donated blood can be used, it must be tested for safety. A donor can donate plasma up to twice a week with a minimum gap of 48 hours and a maximum of 24 times in a year in India. A special technology called APHERESIS is used to draw blood from the patient, separate the plasma out, and return the rest of the blood back into the body. The plasma obtained can be stored in a blood bank. Your whole blood is collected through an intravenous tube in one arm. The blood passes through an apheresis machine which separates the cells from the blood. The cells are collected for storage, and the remainder of the blood is returned through an intravenous tube in the other arm. Convalescent plasma therapy carries the risk of allergic reactions, lung damage and difficulty breathing, transmission of infections including HIV and hepatitis B and C. The third way is to look for drugs to treat. It was found that SARS coronavirus 2's cell entry depends on enzymes ACE2 and TMPRSS2. They can be blocked by a protease inhibitor. Drugs to treat Ebola and related viruses can be used because they may shut down viral replication. Interferon beta, they activate RNase enzyme and degrades the viral nucleic acid strand, but it is a risky method. Monoclonal antibody is a highly effective and safe method. Collect blood from people who have recovered from COVID-19 and isolate the immune cells responsible for producing antibodies. Virus neutralizing antibodies can block the spread of infection. After this current development step, final therapeutic antibody is to be selected, undergo non-clinical or animal study and clinical or human study before production of therapeutics. Fourth way is to find a successful vaccine. There are several vaccine studies worldwide. Some have passed animal tests, some have modified their earlier vaccines, and some are building vaccines based on mRNA of SARS coronavirus 2. RNA-based COVID-19 vaccines. Moderna's mRNA 1273 is a nucleotide-based vaccine. It delivers a genetic sequence for the coding of a spike protein into a host cell, train the immune system to recognize SARS coronavirus 2 spike protein, and produce antibodies required to act against it. Non-replicating viral vector vaccine or recombinant vaccine AZD1222 uses chimpanzee adenovirus which carry genetic material for SARS coronavirus 2 spike antigen. After vaccination, the surface spike protein is produced, priming the immune system. It results in better prophylaxis with fewer doses. Protein subunit vaccine consists of the spike antigen combined with an immunogenic adjuvant to trigger a strong immune response. Whole virus vaccines in which inactivated or killed SARS coronavirus 2 is used. By entering the body, it will create antibody against the infection. When will it be ready? These take time. It may take more than a year, up to 18 months. Usually, on an average, it takes 10 years to develop a vaccine. There are more than 100 vaccines under development for COVID 19 now, but the success rate for vaccine development is only 6%. Yet, vaccine is the only ray of hope in the fight against SARS coronavirus 2. Why does it take so long to develop a vaccine? To produce viral vaccines, candidate vaccine viruses are to be grown in cell cultures. Next, the virus is extracted from the cells, purified, then tested, or they are modified for the specific viral proteins. Then study them in small animals like cats, ferrets. These are usually used for testing antivirals, and hamsters are used here for testing the efficacy of vaccine. Clinical trials on humans take time to check their efficacies and side effects. Phase 1 human trial, the drug is given to a small number of healthy people and people with disease to look for side effects and figure out the best dose to be used. In phase 2 human trial, the drug is given to several hundred people who have the disease to find out whether it works and there are any side effects. In phase 3 human trial, it is a large scale trial. The drug is given to several hundred or even up to 3,000 people. It can take 1 to 4 years to look for how the drug works and to study about the most common side effects. Drugs that are approved for use undergo continued monitoring to make sure there are no other side effects and they are manufactured in large scale. But the fact is, no vaccine gives 100% protection. It gives only 90% protection. Currently, no approved vaccine exists to prevent infection with SARS coronavirus 2. Even in distress, there is hope. And where there is hope, there is a trial. And if you do the work, you get rewarded. It is the result of preparation, hard work, and learning from failure. There are no secret to success. Thank you.